Hi, today's problem going to be about breeding and selection, and in this video I'm going to explain how doing crosses you can find how many genes or alleles would control the trait. And here's a problem. In a rabbit variety 1, ear length average 4 inches. In a second variety it is 2 inches. Hybrids between the varieties average 3 inches in ear length. When these hybrids are crossed to each other, the offspring exhibits a much greater variation in ear length, ranging from 2 to 4 inches. Of 500 F2 animals, 2 have ears about 4 inches long and 2 have ears about 2 inches long. Approximately how many allelic pairs are involved in determining ear length, and how much does each effective gene seem to contribute to the length of the ear? What is the distributions of parent 1 and F1 and F2 probably look like? So if you know how to solve this problem, I always recommend you to stop video here, try to solve the problem on your own, and when you would be ready you can run video again and you can compare your answer with my answer and explanation. So first of all, let me explain what is a additive trait on the simple example from simple Mendelian genetics, but this is going to be slightly modified. So imagine that we have parent 1 that is going to be heterozygous, so going to be capital A small a, and parent 2 also that is going to be heterozygous. In simple Mendelian genetics we would have here two phenotypes, so one would be dominant and another one would be recessive. So this is going to be one phenotype dominant, even these two heterozygous that but would look like dominant homozygous phenotype, and this is going to be second phenotype that would look like a recessive phenotype. But when we have additive traits, the picture would be different. For example, imagine that dominant allele A would stand for the 2 and a recessive allele A would stand for 1. It can be centimeters, inches, or any other characteristic of any trait. So this is going to be additive alleles, and here we would have, for example, 2 and 2, 2 and 1 here, 2 and 1 here, and 1 and 1 here. So as you see, picture would be different. So here we would have one phenotype, and here we're going to have another one that is going to be 3 inches, for example, and here we also would have the third phenotype that is going to be 2. So 2 here and 4 here, and with green color we have 3. So as you see, picture slightly different from what we have in simple Mendelian genetics when we have dominance and when we have recessive pairs. So now we can proceed with our problem, and here is a very simple formula that we are going to use in order to solve this problem, and here it is: 1/2 n squared y 1/2 yn and y squared, and here's explanation. So if you take a closer look, you would see that variety 1 ear length average 4 inches, and second variety is 2 inches, and hybrids between these two varieties average 3 inches. So this is the same picture as we have here. So here would be genotype of extreme genotype of one parent that is going to be 4 inches. Here would be another phenotype of the second parent that is going to be 2 inches, and F1 generation, when we cross these two, would be heterozygous, and that's why we have 1/2 here that is heterozygous and would average between these two extreme phenotypes. Actually, a Punnett square of two parents that is going to be two extremes, or one would be homozygous dominant, would look like this, and another one would be homozygous recessive. And when we cross two such parents, right now I'm only show you one locus with two alleles, so all the progeny would be heterozygous, so would be intermediate, like in the first example, and going to be 3 centimeters, and here for example we would have 4, and here we would have 1, 1, so 2. And as you see, all the F1 progeny, and this is going to be also F1 generation of these two extreme parents. Right now I'm showing you just one locus, and we don't know yet how many locus are there, how many genes, how many alleles controlling this trait, but as you see, this is going to be example of F1 generation, and this would be example of the F2 generation when we took all these genotypes and cross them together. And this is how many different genotypes we are going to see here. And as you see, in F2 generation we have ear lengths that range between 2 and 4, and out of 500 animals, 2 have ears about 4 inches long and 2 have ears about 2 inches long. So imagine if we have 500 animals, and if these animals would have this trait under control of one gene, with one gene represented by 2 alleles, 25% would be of the extreme phenotype, that would be 4 inches, and 25% would be another extreme phenotype that is going to be 2 inches, and 2 out of 500 is not 25%, right? So we clearly see that this trait under the control of more than one gene, more than one allelic pair. So let's find how many allelic pairs control this trait. And we are going to use our formula. So in other words, we can say that 2 divided by 500, and 2 is extreme phenotype. So as you see, we have 2 that is extreme large and 2 that is extreme small out of 500. So we just take any one extreme phenotype and divide by 500, and this is going to be equal to 1/2 n squared. And if we divide 2 by 500, we are going to get 0.0002, that is going to equal to 1/2 n raised to. And now what we are going to do, we are going to take a square root from both sides. So we are going to take square root here and square root here. So on the left side we are going to get 0.0632. That would equal to 1/2 n. So now we need to use reciprocal rule. And what is a reciprocal? And this is very easy. So reciprocal of, for example, 8 would be 1/8, and reciprocal of 1/8 would be 8. Or we also can say that 8 is the same as 8 divided by 1. So we just flip over our numbers, and for example, reciprocal of 5 going to be 1 divided by 5, or if we need a decimal number, this is going to be 0.2. So we can say that reciprocal of 5 can be 1 divided by 5 or 0.2. And vice versa, reciprocal of 0.2 would be 5. And if we multiply both reciprocals, we always going to get 1. So if we multiply 0.2 by 5, we are going to get 1. When we multiply any reciprocals, we are going to get always 1. And this is the rule that we are going to use. So what would be reciprocal of 0.0632? And this is easy to find. We just have to divide 1 by this number, and we are going to find reciprocal. And when we multiply two reciprocals, we are going to get 1. So we have to divide 1 by 0.0632, and we are going to get reciprocal of this number, that is going to be 16.1. And that means that 16.1 equal to 1/2 raised n. And now we can solve for n. So what is the closest integral value for n? And this is going to be 4. So this is going to be 4, and this is going to be our answer. So this trait would be under control of 4 loci, and each loci would be represented with 2 alleles. So total 8 alleles would control this trait. For example, the genotype of parent 1 can be capital A, capital A, capital B, capital B, capital C, and capital D, capital D. And this would equal to 4 inches. And then we cross with another parent that is going to be small a, small b, small b, small c, and small d genotype. And this is going to be 2 inches. And when we cross these two parents, we are going to get F1 generation that is going to be heterozygous for each locus. So this is going to be, the genotype is going to be capital A, small a, and capital B, small b, capital C, small c, and capital D, small d. And this is going to equal to 3 inches. Now, how we can find what is the additive value for each allele? And this is very easy. We just have to divide 4 here, for example, for this parent 1 by 8 alleles, and we would find that each allele would equal to 0.5. So 0.5 for each allele. So I wouldn't write the whole row. And here for the parent 2, we also would have 8 alleles, but 8 alleles combined would give us 2 inches. So here value would be different, so we have to divide 2 by 8, and we are going to get here 0.25 for each allele. So for each allele A, for each allele B, and so on. So combined, such genotype would give us value of 2 inches. And here for the F1 generation, as you see, we would have 0.5 for the dominant allele and 0.25 for each recessive allele. So when we combine all these alleles together, we are going to get 3 inches for the length of the ears in rabbits. And of course, when we cross F1 generation, we are going to get for each allelic pair 3 different values. So many combinations are here, and as you see, using this rule, using this formula, we can find how many genes or allelic pairs each gene in diploid organism is represented by 2 alleles, so how many allelic pairs control a trait. So this is all for today. Thank you for your attention. Please subscribe for my new videos that I post almost every day. Thumbs up if you like this video. Goodbye.